Yeah, so once again, good morning, children. In the last video, we were discussing about whatever the machinery enzymes of the DNA, isn't it? So these all things we were discussed. Also, we were discussed about the Michelson and Stahl experiments. So after that one, we have to continue. There is another important topic with respect to the transcription. Okay, so let us discuss the another important topic, transcription. See, every year in examination in the theory, so either they will ask the question transcription or the translation. Okay, so that's why if you prepare the both the topics, surely one topic will comes for minimum of 5 marks question. Okay, yeah, let's start the discussion with respect to the transcription. First of all, the question arises: how can you define the transcription? While we are discussing the central dogma of the molecular biology, I told you whenever the DNA undergoes self-replication, it will be gives rise to RNA. So that is nothing but messenger RNA. So that phenomena is known as a transcription. So then from mRNA to whatever it will produce is the particular protein synthesis called translation, isn't it? Yeah, so that's what the first process, the transcription. It is a process of copying genetic information from one strand of the DNA into RNA, that too specifically messenger RNA. Okay, did you got it? How can you define the transcription? It is the process of copying genetic information from one strand of DNA into RNA. Okay, so if you observe here the adenine forms the base pair with uracil instead of thymine. So I told you in the beginning of the process, okay, the DNA undergo replication so that it will be gives rise to RNA. So that means the DNA containing the nitrogen bases is nothing but ATGC, whereas whenever it will be get converted into RNA, the adenine forms the base pair with uracil. So that is nothing but uracil. So where it will be uracil with generally? So they are present in the RNA. Okay. So whenever the uracil, the adenine forms the base pair with uracil instead of the thymine, so that's what it will be considered as the RNA. Okay. So I hope you can identify here the base pairs used to make a RNA: A, G, C, U. The beginning from 5 prime to so then up to 3 prime. So I hope you can identify here the template strand and coding strand along with enzyme, RNA polymerase enzyme. Okay, so that you can also identify the messenger RNA. So it is responsible for the synthesis or production of particular protein. Okay, so that's what I hope you can identify here. It's very easy actually, but if you don't understand, it will looks very difficult. That's why. Okay, yeah. And then after what will happen is the process that continue. The DNA dependent RNA polymerase, the name of the enzyme, catalyzes the polymerization only in 5 prime 3 prime direction. Okay, so that the process will continue with respect to the 3 prime 5 prime acts as a template strand, whereas 5 prime 3 prime acts as a coding strand. Okay, so that means I hope you can identify here whatever they have given the template strand and the coding strand, the polymerase, okay, just exactly opposite to the the nitrogen bases. For example, adenine pairs with uracil and uracil pairs with adenine. What about the G and C? Yes, guanine pairs with cytosine and cytosine pairs with uracil. So this is exactly opposite. Okay, so that's what I said. The 3 prime 5 prime, the direction begins from 3 prime to 5 prime acts as a template strand, whereas 5 prime to 3 prime acts as a coding strand. Okay, yeah. So and here you can identify. So that means both strands are not copied during transcription. Remember, even though the DNA undergo replication and DNA will give rise to RNA, but the both strands are not copied during transcription. The question they will ask, okay, justify the statement during the transcription both the strands are not copied during transcription. Why? Like that they will ask the question. Then you should write, during the transcription both the strands are not copied because the code for protein is different in both the strands. Okay, that means the code for protein is different in the both the strands. So this complicates the translation. Okay, so that's why the particular protein, okay, whatever the synthesis, it will be because of the different type of the based on the messenger RNA. So that messenger RNA is responsible for the production of a particular or a different type of the proteins. Okay, so that's the reason. So the code for protein is different in both the strands. Fine. And if two RNA molecules are produced simultaneously, okay, so this would be a complementary to each other. Okay, so that's what it forms a double stranded RNA and prevents translation. Okay, so I hope you can identify here they have given the both the template strand and coding strand. Consider, as I said, if it is begins from 3' to 5', the beginning from ATGC, 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 like that. Okay, but when it comes to the coding strands, as I said, the opposite exactly, the nitrogen bases will be present, beginning from, yes, so that is 5' to 3'. That is TACG, 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 TACG. So now, so when we find like this during the formation of the template and coding, but after the completion of the transcription, now what will happen? So these DNA in DNA nitrogen bases, always binds with the T, right? But after the completion of the transcription, what will happen? In mRNA, uracil replaces the place of thymine. So now in messenger RNA, what are the nitrogen bases are present? I hope you can identify. Yes, 5' to 3' only. All the thymines are replaced by uracil. So now the messenger RNA after the transcription, that is UACG, 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 isn't it? So like that, the thymine replaced by uracil, so then only it will be considered as a messenger RNA. Okay, so this phenomena, so what we said, yes, transcription. Okay, now, so you can identify the transcription. For the purpose of the transcription, so you should know how it will be exactly begin. For the purpose of the transcription unit is very important, so that this transcription unit is responsible for the phenomena of the transcription. Okay, so what is the transcription unit? It is a segment of DNA. Okay, it is a segment of DNA between the sites of initiation and termination of transcription. What is transcription unit? It is the segment of DNA between the sites of initiation and termination of transcription. Okay, then, and what it will be consisting of? The transcription unit, it is consists of majorly 3 regions. Okay, here only they have given the schematic structure of the transcription unit. Okay, just go through it and you can identify. It will be containing majorly 3 regions. Okay, the first one is the promoter region. Okay, the promoter is responsible for the beginning of this process. Okay, so a promoter binding site for the RNA polymerase located towards 5' end, it is also called upstream. And the next one is structural gene. The structural gene, okay, you can identify, yes, where it is present, the region, okay, which is present between promoter and terminator. where the transcription takes place. Okay, so this region is very important for the purpose of the transcription. And the next one is the terminator. A terminator, the site where the transcription stops, located towards 3' end. So that is nothing but downstream end. Okay, so as I said, the beginning from 3' to 5' and 5' to 3'. So this is the schematic structure of a transcription unit. They will ask a minimum 2 marks question. Write the schematic structure of the transcription unit like that. Then you should write this one, and you should explain also. Okay, what is a promoter, what is a structural gene, and terminator gene. It's very easy. A promoter is nothing but the transcription site, the start site, a binding site for RNA polymerase. Okay, so there are number of cases. What will happen in the absence of promoter? There will be no beginning of transcription at all. Okay, so then, and if in absence of the structural gene also, there will be no continuous, the continuity of the process. If there will be no terminator, it is very important. You must have heard about the cancer. Okay, how it will be actually? Yes, consider if any region of our body, physically maybe get damaged, internal or external, how much it is get damaged, that much only cells or tissue should be recovered. Okay, so but in abnormal condition, due to the absence of terminator gene, thus the transcription continues. Okay, then what will happen? There is uncontrolled mitotic cell division. Do you heard of it? Yes. So that is nothing but cancer. Okay, so that's why in a normal, the proper manner, if it will be consisting of promoter, structural and a terminator gene, so the normal process will continue. Okay, fine. And after this one, the transcription unit, as I said, it is a segment of DNA between the sites of initiation and termination of transcription. So generally, as I said, it will be consisting of 3 regions, isn't it? But exactly, if you identify, the beginning from the transcription initiation site, okay, there is a promoter, and if you differentiate, so between the terminator, that is a promoter, and the terminator, where the transcription takes place, that region is a structural genes. For the proper understanding, for the purpose of easiest manner, they have given like this. And the site where the transcription stops, okay, so that located towards the 3 end downstream. So that, as I said, the upper one is a non-template strand. Okay, then the lower one is a template strand, so that we can easily identify the transcription initiation site, the upstream of a gene, and then transcription termination site, it will be considered as a downstream of the gene. Okay, so this is with respect to the transcription and the transcription unit. Okay, so let it be. So in the next class, we will continue with respect to the very important topic, the transcription in prokaryotes and transcription in eukaryotes, because so these are all the topics. If we they discuss at a time, there are number of times they will mix the concepts each other. So that's why whatever I'm doing the particular topics, you should be prepared and thoroughly. The thing is, that particular topic, if you are able to understand clearly, then only it will be easy to study further topics. Okay, so or else it leads to a lot of confusion. Remember, so out of this, whatever the 16 chapters which is present in the second PU, these principles of inheritance, variation, and molecular base of inheritance, these are all the 2 important chapters with respect to the highest marks also. Okay, so that's why you should read intentionally, and while we are watching and while you are reading also, concentrate. Okay, so then only you can understand all these topics. Okay, fine, so let it be. Thank you, have a nice day.